morning and welcome to Global Healthcast, brought to you by Global Health Press. In our special today, we cover pipeline vaccines for children with an update on just licensed vaccines or those which are in phase 3 programs for licensure in countries in Europe. This is a huge topic and I'm not sure if I have all the information needed to be fair and cover all that is out there. So this is why I would be most grateful to receive any additional data, information or comments on any pipeline vaccine from around the globe. Please write me joe.schmitt@globalhealthpress.org. What I will cover today is current vaccines and vaccine development, some basic principles. I will, at the largest part, cover recently licensed and phase 3 vaccines. And finally, I will have a look into the future, what will be needed and what may come. We already have quite a few vaccines out there to be used. We start with maternal immunization and there are 5 products out there. However, none of them is licensed specifically for use during pregnancy. They are recommended to be used during pregnancy, but it is not in the label. That is a difference, and actually the situation will change with the first RSV maternal immunization vaccines coming along very soon. In addition, we have at least 9 vaccines for infants and toddlers, 4 live vaccines, various booster vaccines and vaccines for adolescents and young children, 3 and more for adults, and 9 for those who are 65 and older. In addition to that, we have vaccines that are licensed for special hosts or for special exposures, for travelers, for those with underlying diseases, for those with B and T cell defects. We have vaccines for emerging infections and those against lack of healthcare resource type infections. So this is all already available, and the question is there any more need? And first of all, I would like to show you, yes, the benefit is huge. More than 700,000 deaths have been prevented in the United States alone over a 10-year period using the vaccines that are here on this slide. And in addition, 21 million hospitalizations were prevented and a lot of illness cases as well. So the benefits are huge, and I can promise you, looking into the future, more will come. Why do I think this? The reason is there are new platforms out there that will allow us to come up with more products. We used to have the empirical approach that was sort of slow try and error. We then had recombinant production, which allowed us to give more specific antigens that get a better immunogenicity, memory response, and so on and so forth. Glycoconjugation helped us reduce mucosal colonization and acquisition of encapsulated bacteria, a huge benefit. Then we had reverse vaccinology, which made it more easy to find protective antigens, those antigens on the surface of a microbe that induce protection. And now with mRNA vaccines, DNA technologies, vector technologies, we do have next generation vaccines and platforms that will allow us to produce even protective vaccines against many more microbes. The next slide is from an anti-vaccine lobby website, and there in 2016 they already listed more than 270 new vaccines in big pharma's pipeline. So from what I said before and from this slide, the problem is not to find a new vaccine candidate. The problem is, how can you get it licensed? What you see here is not vaccines. What it in fact is, it is vaccine candidates. They are not licensed yet. So the real issue is, how do you get a license? To find a new antigen out there to protect against an organism is not the big deal. The question is, will you make it to the market? And the hurdles, the barriers are high. The vaccine has to be safe and effective. And how to come up with a new vaccine is shown on this slide. Number one, you need to have a medical need. Number two, you need to find a pathway to license. Even if you have a great idea for a new pneumococcal vaccine that is not based on capsular polysaccharides, how do you want to license it? You would have to do an efficacy study using placebo. And that is ethically not possible because there are vaccines out there that are working and protective, and you would expose subjects to potentially deadly diseases that could be prevented. So you cannot do it. There is no pathway for licensure for a new pneumococcal vaccine that is not based on conjugation technology. I skipped a couple of points, but you need a lot of know-how. And this know-how is not only research, it is also development, it is regulatory, it is intellectual property, it is finances, it is licensing issues, it's tons of things. And this is why Big Pharma has this knowledge and startups don't. And we'll come back to that point later. Most licensures are done and accomplished by Big Pharma and not by startups. The reason is lack of know-how in startups. And finally, you need an investment size that may be more than $1 billion in a situation where only roughly 10% of vaccine candidates ever make it to the market. So you need an investment, maybe up to 10 billion before you find one vaccine that will make it to licensure and will give you a it will give you a return on your investment. Now let's look at this a little bit closer for you to understand the situation. Phase 1 studies. So this is after preclinical development. This is largely done by startups, private biotech companies, some public biotech companies, other pharma companies, and very little is done by Big Pharma. The big new ideas come from outside Big Pharma. If you go to phase 2, Big Pharma catches up. If it comes to phase 3, the other companies largely disappear because phase 3 is very expensive already. It is very expensive because you already need the factory, which is a couple of $100 million if you don't have one that you can use. The final phase 3 study material, the vaccine candidate, has to be produced in this factory to make sure that you have consistency with your production. And registration then, for various reasons, is largely done by Big Pharma. No startups has a representation around the globe to make sure that the vaccine will become a global success story. The success rate for phase 1 is 22%, phase 2 30%, phase 3 64%. And again, the key issues for success are pathway to licensure, global expertises in many fields. This is a plural, this is not one, this is many. And finally, you need billions of euros or dollars to make things happen. Let's go now to the recently licensed and phase 3 products. I looked at the website of these companies and I wrote to all of them, and some of them replied, and I got additional information on their program. So what is coming soon? I think it will be many new COVID vaccines. It will be maternal immunization programs, RSV vaccine, long-acting monoclonal antibodies, new pneumococcal conjugate vaccines, and then shortly I will cover Lyme diseases, dengue vaccine from Takeda, and chikungunya vaccine. COVID vaccines, that has been covered largely. I want to concentrate on pediatrics. What is out there to date is more than 100 vaccines in phases 1 to 3 still in development. 21 have a limited license and 12 of them are fully approved, and 17 have been abandoned for various reasons. So you see, even with COVID vaccines, there's a lot of failure out there. Now, these are the big companies that are globally leading, and you see here the mRNA vaccines. you see inactivated vaccines, you see vector vaccines. Unfortunately, the New York Times stopped recently, end of August, the updates, but you can still find updates on the WHO website. But I think now, September 22, you can still go to this New York website and you will find a very nice update or a situation report on where we are with current vaccines against COVID. Let's focus a little bit on children. Whenever you come up with a new product in medicine that you seek license for, you have to submit a PIP, a pediatric investigation plan. And that means from the beginning of life, from day 0 to age 18 years, you need to have a plan how to get this product licensed for children. So for all of these COVID vaccines out there, already there should be PIPs. What I found is in some countries, there is emergency use authorization for mRNA vaccines against COVID for use in age groups as of 6 months and above. Trials in children I found for children as young as age 3 years, and this is for 2 inactivated vaccines listed here. And there are additional, there are even approved vaccines out there in China for ages 3 to 17 years. If you want to read more about it, you can look here at the WHO statement. Some countries are very reluctant to protect children against COVID because the burden of disease is definitely very small. At the same time, what you have to be aware of is if you don't vaccinate children, they go to kindergarten and infect their kindergarten nurses or their teachers at school. So in the end, these institutions may close, which is a harm to children. So burden of disease only is a very limited view on the benefit of COVID-19 vaccines for children. I don't want to embark on this discussion. I don't have an own view here on my own yet. I still have to see a little bit more data. I just can report now the situation in Europe where we are with pediatric COVID vaccines. We have authorized the BioNTech Pfizer mRNA vaccines. We have the Valneva vaccines, which is not licensed for children, the Novavax vaccine and the Moderna vaccine, the Vaxzevria and the J-Cofton vaccine from Janssen. And the licensures are here from 5 to 11, 10 microgram for Comirnaty, and as of 12 years, it's the adult dose. The Nuvaxovid, that is not licensed for children, for young children, but it is licensed as of age 12 years with the adult dose. And Spikevax is children 6 years of age to 11 years, half of the adult dose. This is where we are with the old variants. And now we see here the new variants, and they are not licensed at all for children under 12 years because only the adult dose is used. And that is true for both Comirnaty and also for the Spikevax vaccine. That is the EMA situation out there. Now, many children have been vaccinated already throughout Europe, and the question is, do these old vaccines, I mean, they are not old in any way, but are these vaccines effective against the new variants? And just one slide on a study done in Portugal, where 98% of the population had been vaccinated, and many of them had already been infected with new variants, with a new variant. So this is a combination of hybrid immunity, number one from vaccines, and number two from the different variants that were circulating in Portugal. And you see here protection against BA.5 if you had received the old vaccines, plus you had been infected with Wuhan Alpha Delta or BA.1, BA.2. And you see protection varies between 51.7% up to 75% if you had been infected with a recently circulating COVID strain. Very good news. So the vaccines are working, and they are particularly working if you had a recent infection with one of the newer variants. Now let's go on with respiratory syncytial virus, RSV. To make a long story short, if you are age 2, you've had RSV here in Central Europe, and I guess around the globe that is true as well, although we don't have data from all countries. But every human being on the globe these days, almost everybody, has had RSV at least once in his or her life. The clinical spectrum is bronchiolitis in infants and other diseases as well. It is a milder disease in children older than 12 months. We see repeated infections in adulthood, and in older adults, you may again see more severe disease resulting in hospitalization. So we have a typically U-shaped curve with a lot of burden of disease early on in life, in infancy, and then again as of age 65 and older, and also in risk subject. So the vaccine need, the biggest vaccine need, would be in infancy and for older adults. I will cover today infancy only. What is the burden of disease? Actually, John McLaughlin at Pfizer did this study. They searched the internet for data on the burden of disease due to RSV in infants. And in the end, from 3,000 studies, 25 were relevant. They're all different types, using active surveillance, using retrospective data, using ICD-9 codes or model-based data. In the end, the summary is the pooled rate is 19.4 per 1,000 infants get hospitalized due to RSV. That is, in other words, 2%, and that is a lot. In addition, 75% and more of RSV hospitalizations are seen in children younger than 6 months. So the burden of disease in children is high, and this is why people around the globe try to find RSV vaccines very, very early on, actually starting in the 1960s. Many attempts were made to find RSV vaccines, but you see this here, they were not successful for various reasons. Actually, Wyeth, later on bought by Pfizer, tried a vaccine in the 1960s, and the result was an inactivated vaccine, and that, if anything, caused more severe disease than preventing any burden of disease. So it was no longer studied, and other attempts to come up with a vaccine against RSV failed as well. Now, why these failures? They are listed here in the orange part of the slide, but the main new finding was really published only in like 10 years ago, 9 years ago, in 2013. And it was the finding that the relevant protective antigen against RSV is the fusion protein. If you want to protect against RSV, you have to induce antibody production against the F protein. Now, the discovery in 2013 published was that the F protein comes in two conformations, pre-fusion and post-fusion. And all past vaccines basically were largely post-fusion antigen vaccines, and they did not work. You see here the difference is huge, and the relevant topics here in color, they are hidden or are in a different conformation, post-fusion versus pre-fusion. And that is the reason why new vaccine developments all of a sudden popped up. And it is unbelievable. There are dozens of companies that are out there developing RSV vaccines because the burden of disease is unbelievably high. You see a lot of burden of disease in infancy, throughout life, and there it is usually not severe, and then it becomes severe again in the elderly, approaching 60 to 120 per 100,000 hospitalization associated with RSV. Hey, let's have a look at what is out there, what we found in publications today. You see different platforms in different icons here. Phase 3, there are 2 monoclonal antibodies out there, one by AstraZeneca and one by Merck. And I didn't find much about the Merck product. And then we have the maternal immunization vaccine by Pfizer, which is a prefusion F protein, and then one by GSK, which is on hold right now for a safety signal. In addition, we have many developments for adult vaccines in phase 3, and I won't cover these today. Many trials are out there, and to get a little bit of an overview, I summarized late developments here on this slide. The nice thing is they all have beautiful names that are either with regard to music like medley, melody, or evergreen, to paintings like Renoir, Matisse, or have nice fantasy names like Grace or RSV. So very, very nice to have this, and this is just for you to have an overview of what will be available or what may be available in the very near future. Let's start with the maternal immunization vaccine that is in phase 3 and that is produced by Pfizer vaccines. The data that I can show you is from a publication, from a phase 2 publication, and what you see here on the right side on top is that you get nice and very high antibodies in mothers vaccinated with this vaccine. The red is the A, and and in blue is the pre-F-B subtype. You get nice antibodies against both. And what you see here, the dashed line, is the limit of quantification. You get also nice antibody titers in infants born to mothers who were vaccinated with this protein. And what you can see here, there is a log-fold higher titer in the newborns than what is observed in the mother. And that has to do with the active transportation of antibody from mother to child after vaccination. Very good news, and no data available yet from the phase 3 trial. Let's see. We already do have data from a phase 3 trial with a long-acting monoclonal antibody. Just for our memory, there is an antibody out there that is used for protection of risk children, that is palivizumab. You have to inject it every month for protection. Now the new product, that gives you sufficient antibody titers for like 6 months. So this is a long-acting monoclonal antibody against RSV, and the idea is you inject it very early on in infancy before the season, and then you are protected during the RSV season. Shown on the slide here is data on top from preterm infants, gestational age 29 to 35 weeks. And on the lower part, you see the respective data from children preterms as of 35 weeks of age up to term infants. The efficacy was 70 and 78% here in preterms. The efficacy against medically attended RSV was 74.5% in this late preterm term infants, and the hospitalization reduction was 62%. So this is the data we already have. The good news is this vaccine, this long-acting monoclonal antibody, has received a positive opinion from CHMP. So it is only a formality that it will be officially licensed in Europe. And I guess we can expect this any day to be out there and be available for pediatricians to be used for the upcoming RSV season. Now, the big question is, what do you want? Long-acting monoclonal antibody, or do you want to have maternal immunization? I think the options are programs with maternal immunization only, long-acting monoclonal antibodies only, or long-acting or universal maternal immunization plus long-acting monoclonal antibodies for preterms only, particularly for those children where the mother was vaccinated late during pregnancy and the child was born very shortly after vaccination, so that there is not much of an antibody transfer from mother to child. So these children definitely would not benefit from maternal immunization. You may also consider giving the long-acting monoclonal antibodies to risk children in addition to any other program, or in addition you could actively vaccinate with an active vaccine children at a later age after they were born. Although, as I said before, what we really want to cover is the first 6 months of life. That is where you see the largest burden of disease. So vaccinating at 3 months or 2 months may already be a little bit too late for optimal reduction of the burden of disease. Which are the key points you should consider for your choice of maternal versus long-acting monoclonal antibodies? I think in a situation where herd protection is likely not possible with any of the current vaccines or long-acting monoclonal antibodies, I think in this setting we have to look at the potential benefits. We have to look at the hospitalization data, the reduction of hospitalization. We have to look at the price, the annual cost, and the strategy that can be used in your country. We have to look into product availability. We've had it several times in the past here in Germany that a product was licensed but not available. We have to look in your country at the time from birth to the time when the long-acting monoclonal antibody is really given. If this is too long and you miss protecting a lot of children because this only happens at age 3 months during the RSV season, it may not be the optimal strategy. You have to look at logistics like cold chain availability, particularly in low middle income countries. And again, implementation. People in GYN do not really always do the best possible to achieve high vaccine uptake, for example, HPV vaccine. In many countries, that is still very low. And the question is, what will be the level of maternal immunization that can be achieved in your country? And finally, particularly for the monoclonal antibodies, you need to have RSV surveillance in place. You have to know if and when the RSV season started so that you can optimally direct and guide the use of the monoclonal antibody. So it's all up to you. You have the choices, and that is always a nice situation if you can choose. Now let's switch gear to pneumococcal conjugate vaccines. And before I cover the vaccines, I want to highlight 2 principles on the biology of pneumococcal diseases. Before a pneumococcus may cause any disease, it has to colonize. That is important because no colonization, no disease. If you are colonized, you have direct infection, otitis media, and pneumonia. Pneumonia is aspiration pneumonia. The pneumococcus is aspirated into your lungs, into the alveoli, and that is how you come up with pneumonia. And then, following otitis media, pneumonia, or directly from colonization, the organism may access your bloodstream, and then you have bacteremia or septicemia, and from there, the organism may go to the brain, to your joints, to any other organ, causing metastatic diseases. But again, no colonization, no disease. The pneumococcal pure polysaccharide vaccines do not prevent the colonization. And that is why they cannot prevent otitis media and they cannot prevent pneumonia. You do have antibodies in the blood, and this is why you prevent septicemia, and bacteremia. However, the efficacy is very low. It is around 50% and lower if you're older age, and it is short-lasting only, like 3 to 5 years, based on data from the United Kingdom. So that is why we need conjugate vaccines. We want to reduce colonization. And if we reduce colonization, we also reduce the burden of disease in non-vaccinated subjects because there is no spread. We induce herd protection. This is a very important concept. Polysaccharide vaccines don't reduce colonization. They are effective against blood-borne disease, but they are not effective against otitis media and pneumonia. Now, another concept you need to know about, the reservoir of the pneumococcus is the young child. And actually colonization rates in infancy and up to age 5 years is up to 50%. Young children infect their parents and their grandparents and other older people. The burden of disease is very high in infancy and very high late in life because the immune responses are low early on and late on in life. And that is why we vaccinate children. We also vaccinate adults, at least in some countries we do. But vaccinating children already protects older adults because of herd protection. You prevent the spreading of colonization if you're using a conjugate vaccine. You're protecting older people. That is another second very important concept. Now, what are the vaccines that are out there? We have a 7-valent, 10-valent, and a 13-valent pneumococcal conjugate vaccines. They are listed here with the polysaccharides of the pneumococcus that are covered, as well as the respective conjugates. And what you can also see is PPV23. That is the polysaccharide vaccine. It covers many more of these antigens that are out there of the pneumococcus, actually 23. The issue is these are not the most frequent ones, or they had not been the most frequent ones in the past before the introduction of conjugate vaccines. Now, very interestingly, the amount of antigen in the conjugate vaccines per polysaccharide is 1 microgram here for PCV10, or 2.2 for PCV7 and 13, or 4.4, or 25 micrograms for the polysaccharide. And this only tells you the polysaccharide is not very immunogenic. You use as much antigen as possible, as much as anyone can tolerate to get an immune response. And again, there is no memory response because polysaccharides don't induce memory. There is no T-cell activation with the polysaccharide vaccination. And protection rate is only very short-lived. It is 3 to 5 years based on data from the United Kingdom. And they can nicely be used as a booster, but for primary immunization, certainly a conjugate should be preferred because then you get the induction of memory cells and the reduction of colonization. Now, this is the vaccines of the future. This is the antigens that are covered, the different polysaccharide. And the golden standard for licensure was PCV13. The new vaccines are the Merck PCV15 and the Pfizer PCV20. In the old vaccine, the relevant antigens were number 3 and number 19A, and PCV13 was not very effective against serotype 3, and serotype 19A, there were nice reductions, but that protection rate there was a little bit slower. So 3 and 19A, they are the really important ones from the old vaccine types. And they are all covered in the new vaccine types as well. And Merck also covers 22F and 33F. And PCV20 covers serotypes 8, 10A, 11A, 12F, and 15B. PPV23 still has more, has additional antigens. But again, the question is, how frequent are they? Are they really relevant? And just to be shown down here again is the amount of antigen for the PCV15 is 2 micrograms and 4 for serotype 4. And again, PCV30 and PCV20 have 2.2 and 4.4. Now the big question is, how relevant are the antigens 22F, 33F, and the others that are in PCV20? I have data here in Europe from Germany, because I'm based in Germany. I have that data from the German National Reference Center for the pneumococcus. And for children under the age of 18 years, what you see here in the light blue is the antigens that are still circulating here up to 2022 that are in PCV7. In gray, you see those that are still PCV13 serotypes. In yellow, you see the PCV15 additional serotypes. And in the darker blue, you see the PPV20 serotypes. And in green, you see the PPV23 serotypes. So there isn't much additional benefit, I guess, from the PPV23. I think in children, no doubt, conjugates are used. They are not even licensed under the age of 2 years. So there is no doubt you use a conjugate. The question is which one you use, and that is certainly up to you. It not only has to do with the coverage, but also with the efficacy against the single strains of all vaccines. So if a new vaccine has a very high activity and protective efficacy against serotype 3 and 19A, that even could be a better choice as compared to a vaccine with a low efficacy against 3 and 19A, if this is covered in a less valent vaccine. In any event, let's look a little bit at adult data. The picture is quite similar. This is again data from Germany. I don't want to go into this. This is data now from the 15-valent vaccine, which is already licensed in the United States for older adults and for children. It's called Vaxneuvance, and this is Vaxneuvance compared to PCV13. What you see is the immunogenicity is higher against serotype 3, and this is post-dose 3. And 19A, it is a little bit lower against 19A. So whatever that means, there is a nice advantage over PCV15 versus PCV13, particularly with regard to serotype 3. Where are we with data from PCV20? The data from a phase 3 study have not been released. I have not seen them at all. What I want to show you is data from a phase 2 study. And again, looking at serotype 3, you see the seroresponders are lower with PCV20 versus PCV13. In addition to the low effect you had before again. And the same is true for looking at the GMTs, looking at the titers. And for serotype 19A, the seroresponse rate are roughly the same, but again, the GMTs are lower for PCV20. Does this mean anything? We don't know. I'm really a little bit afraid of looking at titers only. The only thing that matters in the end is what is the protection rate, and that we don't know. We would only know a couple of years after licensure how these vaccines will have performed in a specific country. It's just the first data we have, and I'm happy to show you here the phase 2 data of the PCV20 and the PCV15 vaccines. And again, the PCV15 vaccines already is licensed for infants in the United States. This is the summary. PPV23, as of 2 years, it can be used, it is licensed, but again, it's not a good idea to use it at all. You should always use one of the pneumococcal conjugate vaccines, PCV10, PCV13, 15 or PCV20. And this is the summary with regard to license. What I showed you is the data for the 3+1 schedule, so 3 doses in infancy, but in Europe, licensure will come for a 2+1 schedule. And their immunogenicity probably will even be a little bit lower than with a 3+1 schedule. So I'm very much looking forward to see the opinion of licensure authorities in Europe and elsewhere on their opinions on PCV 15 and PCV 20 for infancy. Now, again, this is the license in the United States. Both in the United States and in Europe, licensure are based on non-inferior immunogenicity and safety versus PCV 13. That is the benchmark. And again, the schedule is 3+1 in the United States. I would expect there was a 2+1 in Europe. And in the United Kingdom, it may even be a 1+1. That is possible, taking care of a high vaccine uptake and high herd protection. If the burden of disease is low, if the exposure rate is low, you may get away with very little immunity, and there even 2 doses may be protective. What are your decision criteria? And again, this is my own opinion. What do lower titers mean? How relevant is herd protection? And maybe this is more relevant if I relate to the situation in the United Kingdom. What is the effectiveness against serotype 3 and 19A? What is the magnitude of local strain coverage by each of these vaccines against 22F and 33F and the additional antigens in PCV20? And I guess in the end, we will only know 1 to 5 years after licensure. No idea what will happen. I think it is always a good idea to have more than one product out there for your indications, so that you can choose, and if one manufacturer fails, you still have an alternative. So we as customers, we as vaccinees, prospective vaccinees, are in a very good position. There is a lot of competition out there, and we should take advantage of this and make the best choices for us. Having said this, I change gears a little bit and we switch to a sort of rare disease here in Europe, chikungunya, which is in phase 3 and which is produced by Valneva, which used to be Intercell many years ago. Now, chikungunya is spread in many countries in the world, and you see that down here, United States, Latin America, parts of Africa and Asia, but you see also cases identified in Italy and in France. There are outbreaks of disease affecting, 30–75% of the population, and it is difficult to predict if and where such where these outbreaks will occur. What I can tell you is the disease is not funny. The suffering is very high, even if you do not die. So it is a good idea, if there is a high risk, that you get the vaccine. And again, the product is in development by Valneva. It is a monovalent, live attenuated vaccine, and it is based on a strain with a deleted non-structural protein, the replicase complex. So this is the vaccine. It is probably will be licensed at some points for adults and children. Right now, the phase 3 is ongoing with subjects 18 years and older. It is a single dose given intramuscularly. The immunogenicity is descriptive only. There are high titers, I will show you in a minute. There is no surrogate marker of protection to date, but a seroresponder is probably considered to be protected. The duration of protection is certainly not known, but as this is a live vaccine, one would hope that it may be lifelong. The target profile looks very promising to this point. The goal is individual protection, as this is an organism that is living outside humans and has a reservoir outside humans. There will be only individual protection. You will never get something like herd protection. And again, this vaccine may make it to the market shortly, because it has breakthrough designation by the FDA and prime designation by EMA. The data that I can show, again, there will not be any efficacy data because the disease is too rare and you cannot wait for an outbreak. You cannot vaccinate some thousand subjects and wait for an outbreak to come. Then you will never have a product in the near future, and such a study would be immensely costly. And it would be ethically questionable. You cannot vaccinate a whole district in Italy or France waiting for an outbreak to come, right? You cannot do it. So immunogenicity is the basis for licensure. And the immunogenicity is just great. Look here, at day 14, low, medium, and high doses achieve really seroconversion rates that are really 100%. This is really great and very promising. And again, I have no idea if and when the vaccine will be licensed, but it looks very promising. Chikungunya vaccine. Let's go to Lyme disease, and that is a co-development of Valneva with Pfizer. The product out there consists of 3 fusion proteins from 6 Borrelia OSPA serotypes. So 6 proteins were condensed into 3 fusion proteins, and the product is aluminum adjuvanted. The indication will be as of 5 years. The schedule is quite complicated. It is a 2+1 schedule for the first season, 0, 2, and 5 months. And then you will give a booster at 18 months. And the question is, will there be a need for other boosters? The principle, how the vaccine works, is a little bit complicated. You know, the tick with the Borrelia inside sucks blood from a human being. And antibodies against the Borrelia are in the blood of the vaccinated person. Now the blood makes it into the tick, and there the antibodies in the blood kill the Borrelia bacteria. So no antibodies in the blood, no protection. And that is why very easy for this vaccine, no antibodies, no protection. And that is different with many other vaccines, right? In many instances, you have no antibodies and you're still protected. Varicella is one example, or many others. You don't find any antibodies, you're still protected because you had the disease or you had been vaccinated. Here, no antibodies, no protection, because if your blood has no antibodies, it cannot kill the bacteria in the tick. So immunogenicity will be one endpoint. There is a phase 3 study ongoing in the United States and in Europe. There's no correlate of protection. The duration of protection is unknown. There may be annual booster. There may be booster every 5 years. Who knows? The reactogenicity profile looks acceptable. Safety profile looks acceptable. But again, the main point to understand from these vaccines, it kills the target microbe outside the human body. Very interesting concept. And actually, the same was true for Lymerix, which had been licensed by GSK in the United States against Lyme disease and which had been withdrawn from the market some years ago. Let's switch gears to dengue. Dengue has arrived in Europe, as you can see here. In many European countries, it is already established or has been introduced, and in some people consider it as absent to date, but you never know. How can you exclude that it's there and you did not find it, right? So this is the old vaccine that is out there. It is Dengvaxia from Sanofi, a live attenuated chimeric vaccine based on the yellow fever vaccine. And again, it's chimeric with a yellow fever backbone and a tetravalent dengue virus vaccine. The Dengvaxia is licensed in Europe for 9 to 45 years of age, in the United States only until 16 years of age. And that has to do, for the 16 to 45, there's only serology data for licensure, which was acceptable to EMA, but not to the FDA. The vaccine should only be given in seropositive subjects, so you have to do serological pre-screening, and only in positive subjects you can administer the vaccine. That is one of the shortcomings. The efficacy data are shown down here. Again, the duration of protection is not known. Reactogenicity profile appears to be acceptable. There were concerns with the vaccine as a cause of ADE, of disease enhancement related to antibodies, non-neutralizing antibodies. They attach to the virus. The antibody and the virus are then brought into human cells via the Fc receptor of the antibody. And there the virus can replicate in the cell, and then it causes more disease than you would have without vaccination. If that concept applies or not, that is unknown. There are people out there who even say ADE doesn't exist inside the human body. This is a laboratory phenomenon only. But again, I don't want to embark into this discussion. This is the vaccine, and this is how it is licensed in Europe and the United States. Now, let's look at the new vaccine. It is called Takeda TAK-003 dengue vaccine. It is in phase 3. It is a live-attenuated chimeric vaccine, not based on yellow fever, but on a dengue virus 2 backbone. So that is the construction difference, the platform difference. And again, it consists of dengue vaccines type 1, 2, 3, and 4. The target are children 4 to 16 years, no pre-screening before vaccination considered. It's a subcutaneous injection given at month 2 and month 3. There are data from phase 3 available already. It is 72% against hospitalization against virologically confirmed dengue, VCD. Vaccine efficacy varies by age, I'll show you that in a minute, and it declines over time. The duration of protection is not known, but maybe it is only 2 to 3 years. Reactogenicity is profiled. There is no safety concern to date. I show you a little bit more of the detailed data that is published from the phase 3. And again, overall protection rate 72%. If you go to the single serotypes, it is a little bit variable due to low case numbers, particularly for dengue 4. I'm not so much concerned about this. There is also a little bit variation between seropositive and seronegative subjects, and that again has to do with the low case numbers here. If you look at severe VCDs, there is a difference there. So don't be worried about the wide 95% confidence intervals. That has to do with the low case numbers down here. There is one other dengue virus vaccine out there, the Butantan NIH dengue vaccine. It is apparently in phase 3, and it has a dengue virus 134 attenuated platform, plus a dengue virus 2 chimeric strain based on a dengue virus 4 backbone. You see the study details here. This is all I have found, and I have not received any other information on this vaccine. One last vaccine I would like to cover is probably not relevant for Germany, but if you advise pediatric travelers, it may be important to you. It is a newly licensed oral polio vaccine for type 2. And the details are found here in this publication that you can download. You do know that wild-type poliovirus 1 is still circulating. And other than that, we have the circulating vaccine-derived poliovirus 2 circulating in Africa. In New York, actually, there was one case, one paralysis case, and also it was found in the London wastewaters. In addition, there is a circulating vaccine-derived poliovirus 3 outbreak in Israel. So that is the information. So polio is a topic also in Germany. Make sure you've had your 4 doses, preferentially with IPV vaccine. But again, if you travel to one of these CVDPV2 countries, you may need to know about this novel OPV2 vaccine that is out there and that probably will be used in mass vaccination campaigns. That ends our summary of coming soon and phase 3 products. And now briefly, let's think about the future. I think summarizing from what I saw in different pipelines, I think there will be improved new products that are based on available products. Like a monkeypox vaccine, I'm not sure if it will be licensed for children, but it will be developed for adults at least, based on smallpox vaccines. There is a meningococcal ABCWY vaccine, egg-free cell culture vaccines already available for influenza, but that number may increase. Enterovirus 71 vaccine is licensed in China, not licensed in Europe because of different strains circulating, so it may be difficult to license it over here. Malaria next generation, rabies, pneumococcus conjugate vaccine with more than 20 serotypes, and there may be various combinations including influenza and COVID that may come up in the near future. Also, perhaps there will be Ebola, Lassa, MERS vaccines. Group B streptococcus, I would love to see, certainly many developments ongoing. C. difficile, Lyme disease, broad influenza certainly would be nice to have, and you see all of the others here on the list. My hope for the future is really an aspiration, anti-cancer vaccines. I mean, we already have HPV as an anti-cancer vaccine, or the first anti-cancer vaccine, the hepatitis B vaccine. So that is, I guess, work in progress, but much more probably needs to be understood with regard to killing tumor cells. I have taken tuberculosis on the list of my hope for the future, and I scratched it out because I think we have been looking for new tuberculosis vaccines for decades, and we simply do not understand how to protect from tuberculosis. If a person my age comes down with tuberculosis, he has been infected during childhood, and then for whatever reason, your immune system has an immune response that is not protective against the disease. And we don't know exactly what is it that we are not protected. Although we have antibodies and we have all types of immune responses that you can measure in the laboratory, but they are not protective. And as long as we do not understand what protects from tuberculosis, the best thing to do at this point is case finding. And that works nicely. We've shown it all over the world. If you find new cases before one case can infect more than 20 others that are susceptible, the case numbers will go down because of the herd effect. So this is all you need to do. You need case finding. And why is tuberculosis out there? It's crowded living, poverty, war. That is the cause of tuberculosis. And if you stop that, you wouldn't need a tuberculosis vaccine. I hope it's not wishful thinking, but I'm not sure. I have not seen any approach to a TB vaccine that convinced me that there will be a solution to the TB problem in the very near future. In addition to that, I have a couple of things I would like to discuss with you. Number one is the side effect profile of new COVID vaccines. And you see here among rare adverse events, it's acute peripheral facial paralysis, myocarditis, you see immune thrombocytopenia, facial paralysis, Guillain-Barré, transverse myelitis, thrombosis with thrombocytopenia, and others. The big question that I have here, is this specific for the COVID antigen, or is it a class effect of adverse events of the vaccines we're using to fight it? That we don't know. And this is an issue out there that we need to observe, particularly once the case numbers of COVID go down and the disease is globally under control. So stay tuned. This is a big question. The real big question I want to discuss in the end is, why is it we don't have many more vaccines against all the other 250 or roughly 250 organisms that we cannot protect ourselves against? And to understand this, I put up here a slide that was first published by Rino Rappuoli some 11 years ago. And what you see here on the x-axis is the antigen, if it does change or not over time. So there are antigens that never change, every 10 years, every year, every day, like HIV changes the surface every day. On the y-axis, you see what is needed for protection. Antibodies if you're in the top, and T cells if you're at the bottom. Now, most successful vaccines we have to date, they work because the antigen does not change, and they work because antibodies is the protective mechanism, largely. So we need more vaccines, and we need to find better ways to determine the immune response against pathogens that need more than a nice antibody response. And one other item I brought up here is sterilizing mucosal immunity. It would be the third dimension going into the deepness of my room here, and that is sterilizing mucosal immunity. Some of these vaccines, the pneumococcal conjugate vaccine, meningococcal conjugates, and hip vaccines, they already induce mucosal sterilization, and probably so diphtheria and HPV and OPV vaccines. The question is, how do they do this? And we have not understood this. So this is the need for the future, understanding sterilizing mucosal immunity. It may not be IgA. I'm reluctant to say we need IgA, but we don't know the mechanism at this point. So that is the research of the future, I would think. In addition, I think what we need to know is what do we need, what do we want for the future? This is a summary from 1999 from the Institute of Medicine, vaccines that would be a priority at that time, most favorable, more favorable, favorable, and less favorable. And only few of these vaccines are licensed to date. Most have not been licensed. How can we make it happen? How can we get licenses for additional vaccines? And I think what we really would need is someone who wants to invest at high risk. And I think this is available. There are people who are willing to invest money and who are willing to lose $1 billion because they have many of them, and they hope that one of the investments will bring them a return on investment. What we do need is transparent criteria for recommendations and reimbursement. We as a society, you as scientific societies or individual pediatricians, internal medicines, GYN persons, you need to say which are the vaccines you want, which are the vaccines you want licensed, recommended, and reimbursed for your patients. Speak up on this. And then we need national immunization programs that are efficient and that look forward, that have a horizon scanning, what is coming up, where do we need data, so once available, we can recommend these vaccines or not recommend these vaccines, or that we can early on say, the burden of disease is so low, we don't need a vaccine for this. And unfortunately, the latter two items we don't have in many, many countries. We just evaluated 42 countries with regard to their ability to provide excellent healthcare services, and many countries failed to have this horizon scanning. What do we need in the future? This is the CEPI future portfolio: MERS, Lassa, Nipah, chikungunya, Rift Valley fever, COVID-19 vaccine, other diseases. This is we would like to have this. This is the right approach. But again, the willingness to invest may even be there, but you need vaccination systems that embrace this, that come up with the data and that say, yes, we want this. And once it's out there, we're going to recommend it. So if you want to know more about the country vaccination score, you can go to this website. Other than that, I finish for today, and you will be able to download the PDFs of my PowerPoint presentation from below this screen. Thank you very much for listening. I am Joe Schmitt, and I'm happy to be able to present this on behalf of Global Health Press. Goodbye.